Hi students, coming to the next class. In the last class we discussed about the chloroplast, structure of the chloroplast and functions of the chloroplast. Coming to today's class, we are discussing about the ribosomes. See, the ribosomes have the specific capacity, means ribosomes are involved in the protein synthesis process. Whatever that cell is there, the cell contains the ribosomes are there, the ribosomes are involved in the protein synthesis. Ribosomes are involved in the protein synthesis. That's the reason why ribosomes also called protein factories. Ribosomes also called protein factories. Right? See, coming to the history of the ribosomes, first ribosomes are identified by the George Pallade. The scientist name is George Pallade, right? That is about the discovery of ribosomes. And ribosomes are amembranous structures. Ribosomes are amembranous structures. Amembranous structures means around the organelle membrane is absent. Around the organelle membrane is absent. We know the chloroplast, double membrane content structure. Nucleus, double membrane content structure. And the next one is the mitochondria, double membrane content structure, right? And some of the cell organelles like lysosome, right? Endoplasmic reticulum, Golgi complex, right? All these are single membrane content structures. But ribosomes are quite different from remaining all cell organelles. The ribosomes are not having any membrane. Around the ribosomes, not having any membrane. That's the reason why these are called as amembranous structures. Membrane is absent, right? And coming to the next one, ribosomes are made up of with ribonucleoproteins. Ribonucleoproteins. Here ribonucleo means we know about the RNA. Ribonucleic acid. RNA indicates here ribonucleic acid. Ribonucleic acids are combined with the proteins. Ribonucleic acids are combined with the proteins. So when the RNA combined with the proteins, so that case is considered as, that one is supporting to form the ribosomes. Simple. The composition of the ribosomes are RNA plus proteins. Especially we have to know in a cell what are the types of RNAs are present. In a cell, the types of RNAs. For suppose if we see either in prokaryotic cell, either in eukaryotic cell, three types of RNAs are present. One of the RNA is called as rRNA, ribosomal RNA. rRNA, ribosomal RNA. Ribosomes contain RNA is called as rRNA. Next one is the messenger RNA, mRNA. Right? mRNA indicates messenger RNA. Right? See, what is the function of the messenger RNA? See, it passes the message. Or see, the DNA contains sequence. The sequence is transferred into the DNA contain information. Okay, change into the RNA. The DNA contains information change into the RNA. Especially here, mRNA. Here, the message is changing from one form to another form. That is nothing but for suppose this is one of the eukaryotic cell nucleus. It contains the DNA, right? The DNA produces the mRNA. That mRNA transporting from inside the nucleus to the cytoplasm, and it's involved in the protein synthesis. So mRNA binded with the ribosomes. Involved in the protein synthesis. So here what I am telling, the DNA contain message change into the mRNA. Again mRNA transporting from inside the nucleus to the cytoplasm. Finally protein synthesis will be takes place. So messenger RNA, we know the sending messages in WhatsApp, right, or in different apps we have, right? So here message is passing. In biological system also the message is passing from inside the nucleus to cytoplasm by the mRNA. by the mRNA, right? So that is about the mRNA. And last type is tRNA. Last type is the tRNA, transfer RNA. tRNA indicates here transfer RNA. So which one is transferred by the tRNA? See, during protein synthesis, amino acids are required. During protein synthesis, amino acids are required. The amino acids transportation done by the tRNA. During protein synthesis process, amino acids are required. The amino acids are transported by the tRNA. Remember, all these three types of RNAs are involved in protein synthesis. rRNA, ribosomes, directly involved in the protein synthesis, right? mRNA, message passing from nucleus to the cytoplasm. So the mRNA, this is the mRNA. mRNA contains the codons. The codons are codes for amino acids. That is nothing but message. The message is passing from inside the nucleus to the cytoplasm, right? So mRNA involved in the protein synthesis. tRNA, it transfers the amino acids for protein synthesis. See, proteins are nothing but repeated units of amino acids. The amino acid transportation done by the tRNA. Right? So that is about the types of the RNAs. So within ribosomes, rRNA is present, proteins are present. rRNA is nothing but ribosomal RNA. Ribosomal RNA combined with the proteins. The structure is called as ribosome. Right? And these are granules-like structures. We know these are forms the granules-like structures. These are very hard structures, right? See, based on the ribosomes also, we are classified the endoplasmic reticulum. Endoplasmic reticulum into two types. Means presence of ribosomes, granular endoplasmic reticulum. Absence of ribosomes, agranular endoplasmic reticulum. See, presence of ribosomes. See, the ribosomes are granular structures. So that's the reason why the endoplasmic reticulum is called as granular endoplasmic reticulum. Simply we are calling as rough endoplasmic reticulum. Right? Ribosomes are absent, smooth endoplasmic reticulum. Right? Here, right? RNA plus proteins, and next one is the granules-like structures. Ribosomes are granules-like structures, right? Having pores. See, this is one of the, just imagine this is one of the ribosome structure. This is the granule structure. And see inside, see whatever that ribosome is there, okay, the ribosome majorly forms the porous-like structure. Through this porous, the mRNA is moved. Through this porous structure, the mRNA is moved. For suppose this is the ribosome, this is the mRNA, okay, this is the ribosome, right? The ribosome is binded like this, involved in the protein synthesis. Another ribosome binded here, So, ribosomes are having porous-like structures. That porous-like structures are supporting to binded with the mRNA. Ribosomes contain porous-like structures are supported to binded with the mRNA, right? See, these ribosomes are present in the cytoplasm. Especially we know in prokaryotic cells. There is no nucleus, right? Cytoplasm is there. Within prokaryotic cells, 70S ribosomes are there. Within eukaryotic cells, right, there is an 80S ribosomes are there, right? So here, the cytoplasm, either prokaryotic cell cytoplasm, either eukaryotic cell cytoplasm, contains the ribosomes. In prokaryotic cells, prokaryotic cell cytoplasm, 70S ribosomes are there. In eukaryotic cell cytoplasm, 80S ribosomes are there, right? But in eukaryotes, within cytoplasm, some of the ribosomes are binded to the endoplasmic reticulum. Right? That's the reason why that endoplasmic reticulum is called as rough endoplasmic reticulum. Right? So that is about, not only this one here, say, ribosome is also considered as organelle within organelle. We know organelle. Organelle means cell organelles. nucleus is the cell organelle, right? Mitochondria is the cell organelle, chloroplast is the cell organelle. So within the cell, okay, many number of cell organelles are there. See the ribosome, this is one of the important bit. The ribosome is considered as organelle within organelle. Means we know the mitochondria is one of the cell organelle. Chloroplast is also one of the cell organelle. Again, within that mitochondria and chloroplast, another organelle is present. The organelle is nothing but ribosomes. The organelles are nothing but ribosomes. So what I am telling here, one organelle is there. Within that organelle, again another cell organelle is present. That is nothing but ribosomes. We know within mitochondria and chloroplast, there is an 70S ribosomes are present. That's the reason why in eukaryotic cells, both 70S and 80S ribosomes are present. Within eukaryotic cells, both 70S and 80S ribosomes are present, right? So organelle within organelle, right? And the next one, very important one, here the S. Any ribosome indicated with S. S stands for here Swedberg units. S stands for here Swedberg units. I thought I already discussed regarding to the Swedberg units. Again I like to explain here. Swedberg units. Okay, these are also called sedimentation coefficient. Sedimentation coefficient. We know that sedimentation coefficient. See, this is the size of one of the size of the ribosome. This is the small size ribosome, and this is the large size ribosome. So here molecules are separated. Here molecules are separated based on the size and shape. Based on the size and shape. Here molecules are separated based on the size and the shape. So by the usage of density gradient centrifugation, density gradient centrifugation, this is one of the technique. By the usage of this technique, we are separating the molecules based on size and shape. Size and the shape. Here density also there, but majorly size and the shape. So small size molecules, okay, low Swedberg units are there, and large size molecules, high Swedberg units are there, right? See, here Swedberg units are nothing but sedimentation coefficient. Sedimentation coefficient means here molecules are separated based on size and shape, which are subjected to the density gradient centrifugation process. For suppose this is the 30S subunit, the size is small. For suppose this is the 50S subunit, the size is large. So based on the size and the shape, the ribosomes are separated here, right? Small size, okay, here low Swedberg units, large size, high Swedberg units, right? So S stands for here the sedimentation coefficient. I like to say one important point here. See, generally we know that 70S ribosomes, okay, having the subunits are 50S and the 30S. Here 50 plus 30 is equals to 80. Here 80S indicates here 80S. Okay, the subunits are 60 plus 40. Actually 50 plus 30 is equals to 80, right? But we did not mention here 80, we mentioned here 70, right? Here 60 plus 40 is equals to 100, but we mentioned here 80. So why? Why? The reason is, I already explained how many of them remember, I don't know, right? See, this is the large size subunit of the ribosome, 50S subunit, right? This is the overall structure, and this is the small size subunit of the ribosome, 30S. Once both these are binded, see, overlapping takes place. If the overlapping takes place, the surface area is decreased. If the surface area is decreased, the Swedberg units are decreased. The surface area is decreased, the Swedberg units are decreased. Simple phenomena. This is the individual 50S, this is the individual 30S. If the binding takes place, surface area of 50 and 30S subunits are decreased. Once the surface area is decreased, the Swedberg units are decreased. Here also same thing. 60 plus 40, 60, 40, the surface area is decreased. The Swedberg units are decreased to 80. The Swedberg units are decreased to 80, right? That is about the ribosomes. That is about the ribosomes. See, again here the types of the ribosomes we have to discuss here. For suppose the first one, ribosomes, here 70S. See, 70S ribosomes are seen in prokaryotic cells. The molecular weight is 2.4 into 10 to the power of 6 Daltons. Right? Again in 70S, 50S subunits are there, 30S subunits are there, 50S subunit and 30S subunits are there. Right? Here 50S subunit, there is a 23S rRNA, 23S rRNA, 5S rRNA. Right? Within the 50S subunit, 23S rRNA and 5S rRNA is present. And I already said ribosomes are made up of with the rRNA and the proteins. Within 50S subunit, 34 proteins are present. Within 50S subunit, 34 proteins are present. Right? Again, the next subunit is the small subunit, that is the 30S subunit. In that case, 16S rRNA is present and 21 proteins are present. In 30S subunit, 16S rRNA and 21 proteins are present. And next one is the 60S subunit. And next type is, so this is about the 50S subunit. This is about the 50S. This is about the 70S ribosomes. In 70S ribosomes, 50S and 30S. In 50S, 23S rRNA, 5S rRNA. Number of proteins are 34. Both these are combined. Right? And next one is 30S. Within that 30S, 16S rRNA and 21 proteins. Right? That is about the large and small subunits of the 70S ribosome. Right? And coming to the next one is 80S ribosome. Within 80S ribosome, the molecular weight is 4 into 10 to the power of 6 Daltons. 4 into 10 to the power of 6 Daltons. That is about the molecular weight of the 80S subunit. Right? So within the 80S subunit, there is an 60S large subunit, 40S small subunit. Right? So again 60S, there is an 28S rRNA, 5S rRNA, 28S rRNA, 5S rRNA and 5.8S rRNA. Totally here 3 subunits are there. 28S rRNA, 5S rRNA, 5.8S rRNA. Right? Here total number of proteins are 49 proteins. Here total number of proteins are 49 proteins. Right? That is about the large subunit of the ribosome. And next one is small subunit of the ribosome. Here the small subunit of the ribosome, that is the 40S. Here 18S rRNA is present, 33 proteins are present. Here number of proteins are 33. Simple. Here any subunit, it's having two components. One is that rRNA component, other one is the protein. I already said ribosomes are made up of with combination of rRNA and the proteins. Right? So that is about the large and small subunits of the 80S ribosome. So that is about the composition of the ribosomes. Composition of the ribosomes. Then everybody remember within 50S subunit, the 23S rRNA is also called ribozyme. The 23S rRNA is also called ribozyme. See, the ribozyme, that is nothing but one of the important enzyme involved in the protein synthesis. See what I am telling here. Within 50S subunit, 23S rRNA is present. The 23S rRNA is also called ribozyme. Ribozyme is very important enzyme for the synthesis of proteins. In eukaryotes, okay, this is the 28S rRNA. In eukaryotes means here 80S ribosomes are present in eukaryotes, right? Within that 28S rRNA is very important for the protein synthesis. 28S rRNA, okay, this one 28S rRNA, this one is very important, right? So this one involved in the protein synthesis. So in eukaryotes, especially this one is called as peptidyl transferase enzyme. The 28S rRNA in eukaryotes is called as peptidyl transferase enzyme. Right? So in eukaryotes, 28S rRNA responsible for the protein synthesis. In prokaryotes or maybe in eukaryotes, because in mitochondria and chloroplast 70S ribosomes are there, that's why, right? Here 23S rRNA is very important. This one is called as, especially in prokaryotes, the 23S rRNA is called as ribosome, involved in the protein synthesis. Right? And next one, subunits of the ribosomes are there, we'll discuss now. Subunits of the ribosomes are there, we'll discuss now, right? Right, within ribosomes, okay, there is an sites are there, okay, any ribosome, either in prokaryotic ribosome or eukaryotic ribosome, okay, 3 sites are present. For suppose, this is the one of the mRNA structure. Upon the mRNA, during protein synthesis process, upon the mRNA, ribosomes are binded. This is the small subunit of the ribosome, and this is the large subunit of the ribosome. This is the small subunit, okay. Maybe imagine this is the prokaryotic protein synthesis process. This is the 30S subunit and this is the 70S subunit, right? So within that large subunit, especially 3 sites are there. One of the site is called as A site, and one of the site is called as P site, and one of the site is called as E site. I mentioned here 3 sites. The first site is called as A site. A site indicates here whatever that tRNA is there. This is the structure of the tRNA. tRNA comes and binds. Okay, tRNA. This is the structure of the tRNA. This is the amino acid. This is the amino acid. The tRNA comes and bind at A site. So at A site, the A site, okay, which one is binded here? tRNA is binded. tRNA is binded, right? And here A site supporting for the binding of tRNA. And next one, P site. Here P site is supporting for the formation of peptide bond. Formation of peptide bond. P site is supporting for the formation of peptide bond. See, peptide bond is nothing but, see, this is one of the amino acid, this is the amino acid, okay, group of amino acids are there which are connected by the, okay, see, this is the amino acid, this is the amino acid, right? So these amino acids are connected by the peptide bond. Here red color, the line indicates here peptide bond. So many number of peptide bonds are formed for the protein synthesis. The peptide bond formation done by P site. The peptide bond formation done by the P site. Last one, E site. E site indicates here exit site. E site indicates here, yeah. E site indicates here exit site. This is the exit site. What is exit site? See, whatever that tRNA is there, for suppose this is the tRNA binded here, right? And this is the amino acid, and this is another tRNA. This is the another tRNA, right? Binded here. Between these two amino acids, peptide bond formation takes place. Once the peptide bond formation takes place, the empty tRNA, this is the empty tRNA. The empty tRNA comes out. The empty tRNA comes out. So that is nothing but exit of tRNA. That is nothing but exit of tRNA. So this site is called as E site. This site is called as E site. Within ribosomes, 3 sites are there. A site, the very first one, the tRNA binded. It contains the amino acid. P site, peptide bond, right? Forming site. This one is called as P site. Exit site means the whatever the amino acid downloaded tRNAs are there, right? The tRNA, okay, removing out from the ribosomes. So the site is called as exit site. The site is called as exit site. So that is about the different sites. So in cells, different cell organelles are there. In cells, different cell organelles are there. Why ribosomes only have the capacity to synthesize the proteins? Why ribosomes are only have the capacity to synthesize the proteins? The answer is the ribosomes contains one of the important enzyme. The enzyme is called as peptidyl transferase enzyme. The enzyme is called as peptidyl transferase enzyme. Due to this enzyme, ribosomes have the capacity to form the to form the peptide bond. This enzyme majorly forms the peptide bond. That many number of peptide bonds are supporting for the protein synthesis. The many number of peptide bonds are supporting for the protein synthesis. Right? That is about the different parts of the ribosomes. Different parts of the ribosomes. Right? In mammalian mitochondria, we know I already discussed in mitochondria structure. Right? In mammalian mitochondria, 55S ribosomes are present. 55S ribosomes are present. Right? Remember, this one is also very important. In mammalian mitochondria, 55S ribosomes are present. Right? That is about the ribosomes. Next class, we'll discuss about another topic.